Tonight on Nova.
The average time from the kids arriving in the hospital in Promissão until death was 2 hours.
A mystery disease threatens the children of Brazil.
Did Luis Fernando have any illnesses or infections in the month before he became sick?
Follow an international team of medical detectives as they race against time to discover what's killing the children.
Funding for NOVA is provided by Lockheed, a bold new force in systems engineering, management, and technology services for defense, space, and industry. And the Johnson & Johnson family of companies, supplying healthcare products worldwide. Major funding for NOVA is provided by the financial support of viewers like you.
There is a secret fear that haunts the dreams of parents, that in the middle of the night, their happy, healthy child will suddenly and mysteriously get sick, that the sickness will quickly grow worse and worse, and that no matter what they or the doctors do, that child will die.
Which is Rogéria?
This one here.
This one here?
Yes. And this is her sister. And this is her after she died.
In the rural South Brazilian town of Promissão, that nightmare came true. Frightened parents gathered at the local hospital, seeking reassurance. There was an unsettling randomness about this disease. In the same household, one young child would die, and another would be untouched. Even experienced physicians hadn't seen anything quite so rapid and violent.
At first, we thought it might be meningitis. Then we thought rickettsia, or dengue fever, or a number of other possible diseases. But everyone was lost trying to find a cause for what was happening. But the cases kept arriving, and the people wanted to know what was causing them. They were nervous and worried, and some even left the city for fear that their children might get the disease.
News of the deaths in Promissão spread quickly to Sao Paulo, capital of Brazil's most populous state. Children were dying, practically before their parents' horrified eyes. That wasn't supposed to happen in a modern, sophisticated country like Brazil. In this teeming city of 16 million, no one felt safe. At the Adolfo Lutz Institute in Sao Paulo, Brazil's premier medical laboratory, scientists took samples of blood and tissue from the dead children. Growing out the microorganisms they found onto culture plates, they looked first for meningococcemia, a severe bacterial infection that often causes meningitis. But the tests were negative, leaving no clear second choice, nor any idea of whether it was bacterial, viral, toxic, or something else. In the meantime, more children were dying. 4,000 miles to the north, in Atlanta, Georgia, is the headquarters of the Centers for Disease Control, the division of the Public Health Service charged with monitoring disease outbreaks in the United States and around the world. The Brazilian case ended up on the desk of the Special Pathogens Branch, which is often called upon to investigate mystery diseases.
I got a telephone call one day from Emil Gottschalk at the Rockefeller, and he said, a friend of mine from Brazil has just called, and they've had a very strange cluster of severe disease. I think you should get in touch with them. It was disease of unknown etiology that had a very high fatality rate. And one of CDC's mandates is to investigate diseases of unknown etiology. We were also concerned because the city of Sao Paulo has 16 million people, and even though there had only been 10 cases in Promissão, if the disease reached Sao Paulo, there was potential for much greater morbidity and mortality. It was impossible to get accurate information about what was going on from Atlanta. So we sent one of the EIS officers, David Fleming, to investigate.
Each year, CDC sends investigators to over 100 countries of the world. And the reason is, we really do live in a global village where there are no divisions between countries, and where the speed of travel from one country to the next exposes us to what happens in Zaire or in Brazil or Cameroon. And so if we really want to protect, even if we were totally selfish, if we simply wanted to protect Americans, we have to know what's going on around the world in order to know what to expect here.
David Fleming was in the second half of his 2-year stint with CDC's Epidemic Intelligence Service when he went to Brazil. The EIS takes young doctors or other health professionals, gives them a few months' intensive preparation, then sends them out to face public health threats, some of which have never been seen before. So, armed with the EIS officer's red cooler of lab supplies, Dr. Fleming arrived at the Adolfo Lutz Institute.
It was difficult at best to figure out exactly what was happening, but the information that we were getting said that there were about 10 sick kids who became ill with fever, vomiting, abdominal pain, developed purpura, which is bleeding under the skin, and then died. Cultures that they said they had obtained were negative, and it was unclear what was happening. Hi, I'm Dave Fleming from CDC. Am I in the right place?
I'm Christina.
Christina, nice to meet you.
You're
Vera, nice to meet you.
Christina.
Oh, can we? There's a lot of things that needed to happen right away. The first and most critical thing was that I needed to be brought up to speed because the Brazilians had done an awful lot of work before I got there. And so we spent the first 3 or 4 days really educating me what was known and what had been done already. Nice to meet you. At some point, what we need to do is to make a complete listing of all the specimens that you've gotten and exactly what viruses you tested for, what bacteria you tested for, so we can review it.
We couldn't find any organism from these samples.
What specifically did you look for in the blood cultures?
We looked for Neisseria meningitis, Haemophilus influenza, type B, and pneumococcus.
Strep pneumo?
Yes.
Was that antigen detection or was that culture?
By culture. And
We then sat down and went over the clinical, the epidemiologic, the pathologic results of the cases, and one by one, we were forced to scratch out all of the things on our list, leaving only unknown infectious agent, unknown environmental agent, unknown immunologic reaction.
The Brazilian medical team didn't know what was causing the disease, but they knew what was causing the deaths. In each case, autopsy reports described a collapse of the body's vascular system, causing blood pressure to drop rapidly and blood serum to leak into the tissues and lungs. The victim would go into shock, unable to stabilize body functions, bleeding to death internally and drowning in his own fluids.
Stomach? Do you have any brain?
Kidney.
Kidney? Skin?
Skin.
We spent 3 or 4 days talking with the epidemiologists that had been out to Promissão, the clinicians who had taken care of the kids, the pathologists who had done some of the autopsies. And in going through it, became more and more convinced that we really didn't know what was going on in Promissão. and that we were back to ground zero, if you will, and that the critical thing to do was to go out to the town and start from scratch.
In contrast to the giant, bustling city of Sao Paulo, much of Sao Paulo state is rural and agricultural. In recent years, coffee growing has given way to the production of sugarcane, which is mainly processed into fuel for Brazil's alcohol-powered automobile engines. Sugarcane is the largest single economic activity in all of Sao Paulo state. Everyone who lives in the towns that border the fields is in some way or another dependent upon this industry. Promissão is a typically peaceful farming town of about 20,000, 6 hours' drive from the city of Sao Paulo. There was nothing to distinguish it from scores of other such towns in the region, except that for some reason, children between the ages of 3 months and 9 years were dying here. Fleming arrived at Promissão General Hospital, accompanied by an epidemiologist from Sao Paulo who had been working on the case since the beginning. He carried with him the same fears that haunted his Brazilian colleagues. Were they seeing the beginning of the next plague, the next cholera, or even the next AIDS?
One of the most critical things that we had to do in Promissão was to meet with the clinicians, the doctors, who had cared for the cases, because they were the people who knew best what the clinical symptoms were. They were the ones that were most likely to see things that might have linked the cases with one another. The docs in the town had been through a lot. They were accused by the town of being incompetent because these children had been dying. They were frustrated because the children had been dying and there was little they can do about it. Everyone was concerned about potentially there being more cases. So it was a difficult situation.
By the time Fleming arrived, the outbreak seemed to be over. There hadn't been any new cases for more than a week. But the medical community still lived in terror that this scourge, which had begun for no reason and ended for no reason, would begin again. The only thing that linked the cases together was that all of those afflicted were children, intensifying the sense of guilt and helplessness in parents and doctors alike.
In medicine, what a physician is trying to do is to bring the full body of knowledge that exists for a particular problem and apply that knowledge to an individual person. In epidemiology, one is trying to figure out what's happening with a disease that involves a community, and you're looking for the distribution of that disease, the determinants or what caused it, what are the effects of that disease, and can you make a diagnosis of what the disease is doing in a population and then intervene to change the disease.
Is it?
An epidemiologist works on all those things.
This one?
Yeah, this one. I still this way.
Yeah.
There is an established format and procedure that you use to investigate an epidemic. And an epidemiologist is very much like a detective. You dig. You ask a lot of questions. You interview all of the victims. Or if you can't do that, the closest witnesses.
Tell me about when Tatiana became sick.
Quando a Tatiana ficou doente, o que aconteceu?
Seus sintomas? Ela só teve febre.
Fever. And when did that start?
Na parte da tarde.
De
tarde.
The afternoon. Before the fever started, did she have any other symptoms like nausea or vomiting or anything like that?
Antes da febre iniciar, ela teve algum sintoma, como náuseas, vômitos?
Não, só teve febre.
Just fever.
In the month before Tatiana became sick, did she have any other illness or infection? The first thing that you need to do is to invest a lot of time in talking in detail with those who know best what happened to the kids, and those are the parents. So the critical thing that we wanted to accomplish in Promissão initially was to sit down and have them tell us, as best they could remember, everything that had happened to their child in the month before they became ill. One of the things that was most striking about this disease was the story of how it happened, and that was one of the aspects that we really wanted to talk to the parents about. The typical story was that you had a young child who was healthy, well, playing with his or her friends, ate dinner, the mom put the child to bed. In the night, she might have heard him cry, got up, checked, maybe felt a mild fever, didn't think anything about it. The following morning, when she walked into the child's room, she heard the child whimpering and looked, and the child had just a few little red spots on their arm. But she was enough concerned because the child had a little bit of vomiting and some fever that she'd take the child to the hospital. In Promissão, nothing is very far from anything else. But the consistent story was that on the way to the hospital, she would see more and more red spots appearing on the child, such that by the time the kid got into the emergency room, basically their arms and their legs would be covered with this bleeding under their skin that was rapidly coalescing into larger bruises. The child at that point would become distinctly sick, shocky, and the physicians would throw up their hands because it was clear that despite anything that they were going to be able to do, that child was going to die. The average time from the kids arriving in the hospital in Promissão until death was 2 hours. First, we noted that most of the cases had received an immunization in the 2 months before they became ill, and so there was some speculation that maybe this was some sort of immunologic reaction. Second, most of the cases lived in the outskirts of the town, near the sugarcane fields, and so we were thinking perhaps there was some vector, a mouse, a rat, a fly, that had come in and somehow infected the children. In addition, there were a number of theories circulating as to potentially what could have been causing this illness, ranging from pesticide spraying that had been going on when the outbreak was occurring, burning in the fields, and some environmental toxin.
Of all the environmental factors, the one that caused the most concern was the burning of the sugarcane fields. Farmers would burn off the undergrowth to facilitate harvesting the vast expanses. The burning and subsequent refining left the air heavy with the cane's sweet residue. Did this have something to do with promoting the disease? Did it attract insects or other potential carriers of microorganisms? The Brazilian investigators were grasping at any clue. Was the disease an ironic consequence of a recent inoculation campaign? Or did it have anything to do with an infection called conjunctivitis, commonly known as pink eye, that broke out in the town a few weeks before? None of these clues seemed promising, but they all needed to be checked out.
Sou Maria Lúcia, Dr. Fleming. Ele quer saber quando que o Luiz Fernando ficou doente, o que aconteceu exatamente.
Bom, foi numa segunda-feira, né?
Well, it was on a Monday. He played the whole day, and in the afternoon I gave him a bath. Right after his bath, he had a fever. He had the fever all night, a fever that wouldn't go away. I gave him aspirin to lower the fever, but it wouldn't go down. The fever would not go away. In the morning, I took him to the hospital, and he was all purple. And all he did was cry, "Mommy, Mommy." He was really sick. We took him to another hospital along with a nurse, and when we arrived, they yanked off his clothes and put him in an oxygen tent. At that point, foam started coming out of his mouth, and he got more purple. It was like he had blood between his meat and his skin. I was in the room with him, and the doctor asked me to leave so I wouldn't see. And right after, the doctor came out and said he had died. It was really fast. The disease didn't even last 24 hours. It was really fast.
Did Luis Fernando have any illnesses or infections in the month before he became sick?
Está perguntando se o Luiz Fernando teve alguma doença mês antes dele ficar doente.
Não, nenhuma.
Did he have any history at all of conjunctivitis in the month before he became sick?
Ele teve alguma história de conjuntivite mês antes de ficar doente?
Teve conjuntivite, mas foi uns dias antes, né? Poucos dias antes.
Did any other people in your house have conjunctivitis during the week before Luis Fernando was sick?
Ele pergunta se alguém mais da sua casa teve conjuntivite na semana que o Luiz Fernando ficou doente.
Não, ninguém. Ninguém da família.
Não.
Não teve.
From most of the parents, we got a history of a preceding purulent conjunctivitis in their child. Now, conjunctivitis is a mild eye infection. It's as common in Brazil as upper respiratory infections are in the United States. It was something that the parent almost left out of the history. But we did hear from most of the parents, oh, and 2 weeks before my child became ill, he was sick with conjunctivitis for a day or two. I didn't think anything more about it. It's a well-known phenomenon in epidemiology that when you have a severe disease and you talk to parents or loved ones, that they're very likely to remember inconsequential illnesses that occurred beforehand. And so I think my bias going in was that this conjunctivitis story that we were getting was likely to be just because the parents were bereaved and they were trying to remember every single possible little thing that could have happened to that child. I particularly believe that when I got to Promissão, and of the first 50 kids that we saw running around in the streets, at least half had some sort of conjunctivitis. It's very, very common. But it's the job of an epidemiologist not to make those judgments, right? But to let the information, let the science tell you whether or not it's significant. And the way that you do that is by comparing the information that you get from the cases or the parents of the cases to similar information that you get in exactly the same way from the parents of children who did not become ill. So that was the next step. After we talked to the parents and developed a couple of potential things that might link the cases, was to identify a subset of well children in Promissão, go out and talk with their parents, and see what kind of history we got from those parents.
Using local vaccination records, the investigators began setting up a case control study, which is at the heart of the science of epidemiology.
These are the immunization records?
São os cartões de vacinação?
São todos esses cartões de vacinação.
How are they arranged?
Como eles são arranjados, os cartões de vacinação?
Em ordem alfabética.
The alphabet order.
Is there any way to pull just on kids who were between 3 months and 12 years old?
Ele queria saber se tem uma maneira das fichas estarem arranjadas para se poder coletar crianças entre 3 meses e 10 anos.
Sim, através do outro fichário.
Over there?
Over there.
Okay.
Epidemiology is the scientific method that we use to see how diseases are transmitted. It's different than the usual scientific method. You can't go into the laboratory and perform a nice experiment where some people get the disease and some don't. You have to use the observational data, what has occurred, to try to study why that occurred.
In a case control study, the investigator wants to identify a group of individuals that matches up with the victims of the disease as closely as possible in every way, except that they did not become ill. The next step is to formulate questions that will help determine the variables that distinguish the two groups from each other, and suggest reasons why one became sick and the other didn't. The case control study would take several more precious days. And though fearful parents were anxious for quick answers and reassurance, Fleming knew this was the only hope for finding out what had caused the children's illness. Over the next several days, the epidemiologist interviewed scores of control subjects. By comparing the two groups, they were able to rule out immunizations, animal carriers, and environmental toxins.
The thing that did distinguish the children who became ill from the well children was that the children who became ill did have this history of conjunctivitis, and we did not get that same history in the children who remained well. Sure, we heard about a little bit of conjunctivitis, maybe 10-20% of the kids, but nothing like the 90 to 100% that we were seeing in the kids that were sick.
I first recall as a medical student having a house officer say that when you hear hoofbeats, you should think of horses, not zebras. That is, if you see a person with a certain symptom complex, you should think of the common things, not the uncommon things. Damon Runyon once said that the race is not always to the swift, but that's the way to bet. And so that's going to be what you usually see. The common things are the common things. But in public health, while we use the same expression, I think there's an advantage to the young Epidemic Intelligence Service officer who goes out, sees a problem, he thinks of horses, and once he decides he should look beyond that, he thinks of zebras, and he comes up with things that would not have been thought of by other people.
Boy, the heart really goes out for...
The clue that emerged from the case control study was the conjunctivitis, an extremely common infection, but so mild that any link to the deadly mystery disease seemed implausible. While they were still analyzing results, the investigators learned that a disease that sounded like it could be Promissão fever had broken out in the town of Londrina a few months before.
We went to that second town. We went to Londrina, and we repeated the study that we had performed in Promissão. We identified the parents of children who had become ill and asked them about a history of conjunctivitis in their child, and we got it.
It was now clear the disease could strike anywhere. With a heightened sense of urgency, the investigators honed in on their pursuit of Brazilian Purpuric Fever, BPF.
It was at that point in the investigation that I personally came to believe that there really was something going between conjunctivitis and Brazilian Purpuric Fever, that there was some link between the two. The question becomes, what is the nature of that link?
Conjunctivitis can be caused by a number of different microorganisms, including various strains of virus and bacteria. Since the infection was rampant in the area, it was possible that BPF might be close behind. So it became crucial that the team collect both control samples and case samples to analyze in the lab. They took their control samples from the eyes of randomly selected children with conjunctivitis. Then they needed samples from the actual victims of BPF. But by the time those children had died, their conjunctivitis had already cleared up. So the investigators took samples from relatives and close contacts. If these individuals were infected, they probably had the same conjunctivitis-causing strain as the victims, but for some reason had not developed the fatal disease.
So now we had a population of bacteria and viruses that were associated with conjunctivitis in Brazil, some of them potentially with this disease, some of them not. And that gave us something to work with finally, because now we could take those samples back to the laboratory. One of the most frustrating things about this outbreak was the cases had occurred before I got there. And so I hadn't seen one, and I really didn't know clinically what we were dealing with. Now, that's good because everyone wants the epidemic to be over, but it was bad because in order to really understand this mystery disease, I felt as if we needed to see a case.
Fleming had been in the countryside about 3 weeks. With no active cases, it was time to return to Sao Paulo. In the history of medicine, almost all progress is preceded or accompanied by tragedy. This time was no exception. The very day Fleming left for Sao Paulo, the next child became sick.
Nós tivemos caso que levamos para São Paulo.
We had a case that we took to São Paulo in an ambulance. There was a nurse, a driver, and me. Midway through the trip, as we realized the child Rodolfo was getting worse, we asked the highway police to buy more antibiotics for us and that they be delivered to us at the next highway police station in Bauru. We were able to make the 6-hour trip in 3.5 hours. In spite of the increasing doses of antibiotics we were giving him, we noticed that his petechiae were increasing. And as the minutes and hours passed, the petechiae kept getting worse. When we got to the main highway, we noticed that there was necrosis of his nose and earlobe. And whenever that happened, it was a sign that it would be difficult for the child to survive.
The sick child was transferred to the main teaching hospital in Sao Paulo, and as soon as he arrived, we rushed over there. And I can remember walking into the room in which he was and being blown away by the severity of this illness, because there was this young child, 2-3 years old, who I knew from the story had been perfectly well the day before, lying there, obviously in shock, with purpura, which is bleeding under the skin, his arms, his legs. His nose had turned almost bluish black. His ears had turned almost bluish black. And it really became clear that despite receiving first-rate Western world medical care, that there was nothing that we were going to be able to do to save this kid. And we spent the next couple of hours really standing over this child, watching him die.
Two days later, Fleming met for the last time with his Brazilian colleagues.
Okay, we can say we know a couple of things about the conjunctivitis. I think we can say it's infectious, transmitted person to person, it's purulent, it's got a low attack rate, affects mostly kids, so we think it's bacterial. What am I leaving out?
Infeccioso mais comum isolado das conjuntivites purulentas foi o Haemophilus aegyptius.
I'm sorry, I didn't follow that.
The most common microbial agent was
H. aegyptius.
Haemophilus aegyptius.
Among the strains of conjunctivitis that were collected in the field, the one organism that seemed to be associated with the cases was a bacterium called Haemophilus aegyptius. This left the team with a perplexing contradiction.
We're back to the laboratory now.
The epidemiology indicated that the cause of BPF might be this organism. But Haemophilus aegyptius is a common bacterium never linked to serious disease.
We can both work on it together.
So the mystery remained as baffling as ever. After 5 weeks of investigation, David Fleming left Brazil. He carried back the Haemophilus aegyptius samples gathered in the field and tissue specimens taken from victims, including Rodolfo, the boy he had seen die. Could the laboratories at the Centers for Disease Control, the same laboratories that had successfully cracked Legionnaire's disease and so many other medical mysteries, help solve this one too? When Fleming arrived back at CDC, he shared his suspicion that there was some link between the benign Haemophilus aegyptius bacterium and the deadly Brazilian fever.
That was met with a considerable amount of skepticism. It turns out it's one of the first bacteria ever described way back in the 1800s. It's been known for 100 years. It's a common cause of conjunctivitis in third world countries, and never, never had it been associated with anything more than a self-limited eye infection. So for a time, we were all walking around thinking, great, we've discovered 100 years late that Haemophilus aegyptius causes conjunctivitis, and we thought we must just have missed whatever organism it was that was causing the conjunctivitis that caused this more severe disease.
As soon as David got back, we needed to plan a course of action to try to find out how Haemophilus aegyptius was involved, or whether other agents might also be important. What we did was we got together a group of laboratory scientists from our own group and also from other areas at CDC to hear what had happened in Brazil and to plan what would be done next. The group was convinced by the association with conjunctivitis, skeptical about the role of Haemophilus aegyptius, and thought that we definitely needed to do further work to rule out other known agents.
We have some conjunctival strains, primarily Haemophilus aegyptius, but also some other organisms isolated from the towns where the disease was occurring, as well as from some control towns. And then there's that one non-sterilely obtained Haemophilus aegyptius isolate that was obtained from a case shortly after death.
We're still trying to be sure we're dealing with a bacterial infection. It could be viral, or it could be some kind of immunologic response. And I think the path specimens may help us with that.
If you get me the tissues, I'll evaluate them for any histopathology. And as a matter of fact, we can do a battery of special stains looking for a particular organism. And if H. aegyptius is there, you might be able to pick it up in the tissue somewhere.
One thing that's important, I think, is that it's still unclear whether this is one illness, and so we need to be looking for unifying features in the pathology.
We were left with a feeling that looked like Haemophilus aegyptius might be involved, that it had caused the conjunctivitis that preceded the illness, but we couldn't explain how this severe illness could have been caused by Haemophilus aegyptius.
Among the strains that way.
Dave, were the serum samples...
Dr. George Carlone was one of the key laboratory investigators for CDC.
The epidemiologists are charged with identifying the disease, evaluating the disease, and bringing back appropriate specimens to the laboratory. The laboratory, on the other hand, using all the information that the epidemiologists can obtain, analyze the samples, compare the samples, and come to a conclusion.
While Carlone and Fleming analyzed the conjunctivitis from Brazil, other CDC laboratories began studying the blood serum and tissue of victims, looking for agents more likely to have caused death in the children. The virology lab hunted for evidence of virus and related organisms, while the bacteriology lab searched for hidden bacterial strains. Pathology examined the autopsy specimens to obtain clues suggestive of unusual infections or toxins. The immunology lab checked for allergic reactions or environmental agents. But after weeks of work, no one had come up with anything. In the meantime, Fleming and Claire Broom contacted Haemophilus aegyptius experts around the world to see if they knew anything that might account for the deaths in Brazil. The response, over and over again, was no.
That would let us nail Haemophilus aegyptius.
There was a potential alternative explanation, and that was, in some way, the Haemophilus aegyptius was associated with this disease, but maybe it was a certain type of Haemophilus aegyptius that was different from all the other types. The only way to figure that out at that point is by shotgunning it. By that I mean giving the isolates that we had cultured from these kids to various laboratories and saying, we're not going to tell you which of these specimens were associated with this disease and what weren't, but we want you to try to figure out ways that you can distinguish these isolates from each other.
This technique is known as a blinded study. The identity of each lab sample is unknown to the investigator to eliminate bias. It isn't until the end of the experiment that he is told which are the disease strains and which are the control strains. In addition to the control samples of Haemophilus aegyptius gathered in Brazil, Carlone added strains from CDC's own collection, stored here for this type of experimentation.
We compared these strains to the organisms that we had previously stored, And what we found is that the strains that David brought back were very similar, both biochemically and genetically, to the organisms that we had stored previously. At this point, that really didn't tell us much. Also, people were still very skeptical, maybe even more so at this point, because there was no differences. These were organisms that had been found throughout the world. They had never caused this really horrendous disease. The clinical symptoms have never been associated with this disease, and the skepticism was still there.
With the first set of tests showing no differences between the case strains and control samples, Carlone's team probed deeper into the organism's genetic material. They were looking for one of the most common characteristics that distinguish similar bacterial strains from each other: plasmids. Plasmids are circular pieces of DNA that lie outside the main strands of genetic material in some bacteria. Not all bacteria have plasmids. Others have more than one. Plasmids are generally benign, but in some bacteria, specific genes on the plasmid enable the cell to produce toxins, which can cause serious illness or even death when released into the bloodstream. To determine whether an organism contains a plasmid, scientists use a technique known as agarose gel electrophoresis on the bacteria's DNA, which visualizes the plasmid as a band on the gel.
What you got?
Well, I took the 14 strains that you gave me, Dave, and I ran plasmid profiles, and I have the results for you.
Hang on. Okay.
By the way, all 4 of these plasmids were 24 megadalton plasmid, and the best that I can tell, they're all the same molecular weight.
Okay.
When Carlone's lab completed its test for plasmids, Fleming returned to identify the sources of the various test strains.
Negative. 1950 was positive. 1951 was positive. 1960 was negative. 1963 was negative. 1965 was negative. 1949 was positive.
49 was what?
1949 was positive. 1975 was negative. 1980 was negative. 2000 was negative. And 2001 was negative. So out of the 14 strains you gave me, only 4 of them were positive for the plasmid, Dave.
That's really interesting. The 4 positive ones are the 4 case-associated strains from Brazil. The others were control strains. The strains with the plasmid were the strains that epidemiologically were associated with the severe disease down in Brazil. It was that point, I think, that everyone became convinced that maybe we had something here.
Now, there is an additional analysis.
Once they knew that the disease-associated strains contained a plasmid, they had to see if it was different from all the other plasmid-containing strains of Haemophilus aegyptius. To do this, scientists use an enzyme that will cut the plasmid's DNA only where it recognizes a particular chemical sequence. Where the plasmid ring splits will distinguish one strain from another, and the individual pieces will make a distinctive pattern of bands when flattened out on a gel.
The way you look at these things is by running the DNA material through a gel and staining it and photographing it with a UV light.
Like fingerprints left at the scene of the crime, the pattern revealed here was distinctive and unique, and never before associated with Haemophilus aegyptius.
Here's a picture of the enzyme digest of the strains from Brazil. This is what came to be called the case clone, which you can see has this pattern of DNA. And these are other control strains from Brazil and from the United States, which clearly have different patterns in their enzyme digest.
The conjunctivitis samples from Brazil had revealed a new mutant strain of Haemophilus aegyptius, characterized by this particular genetic pattern. But to confirm that it was the cause of the deaths, scientists would have to isolate this bacterium in the blood of the victim. Only then could it be proved to have passed from a mild conjunctivitis to a systemic and fatal disease. Information on the mutated bacterium was sent to Brazil, so epidemiologists there would be on the lookout for the new strain. Events soon gave them the opportunity. The killer had struck again. Samples isolated from the stricken children's blood were sent back to CDC.
When we got more strains in from Brazil as the epidemic progressed, we found that there were a number of strains which had the same plasmid band.
This evidence showed that a new mutated form of Haemophilus aegyptius had in fact killed the children in Promissão. It was dubbed the BPF clone and proved the killer was still on the loose.
Since we knew how devastating this disease was, we tried to work on methods for rapid diagnosis. It was important to, and still is important, to identify those individuals that may in fact be infected with the BPF clone.
The Brazilian and American teams tried many diagnostic techniques to rapidly identify the organism under field conditions. Cristina Brandaleone at Adolfo Lutz found the one test that proved effective, a well-known technique called slide agglutination, a blood serum test keyed to detect specific microorganisms. In 24 hours, any suspected new cases could be identified while they were still at the conjunctivitis stage. Before the test could be implemented widely, the disease broke out again in the town of Serrana. Now there was a way to identify the deadly strain. The next question was, could it be treated? Since BPF is caused by a bacterium, it seemed likely that if caught in time, during or shortly after the conjunctivitis phase, the disease would respond to antibiotics, and lives could be saved. David Fleming had completed his term with the EIS, so CDC sent Lee Harrison, a physician fluent in Portuguese. He was accompanied by Lígia Regina Querpontes, an epidemiologist from the Regional Health Center of Ribeirão Preto. And this time, the medical detectives had a new element to work with: survivors. Marlon Marquez Lima was extremely ill with BPF when he was brought into the emergency room. But Brazilian health authorities had issued alerts and instructions to all hospitals. A rapid diagnosis by informed, vigilant doctors and an intensive course of antibiotics saved his life. In other cases, only more dramatic intervention could fend off death. The course of Simone Poiares de Camargo's illness was so violent that massive antibiotics were not enough.
She was really sick. She was black and her body was all swollen. The doctor told me that she had 12 hours to live, that she was not going to survive. She couldn't take it. She was going to die. But thank God she made it.
In a heroic effort, Simone's doctor saved her life, but at the cost of her leg. 5 other children were not so fortunate. But the Serrana outbreak showed that with early diagnosis and treatment, the odds improved dramatically.
In the investigation of the cases in Serrana, it was possible to look at children, to look at the antibiotics which had been given to children and find out that children who'd received antibiotics early in the course of illness were less likely to die than children who received antibiotics later in the course of illness. And this suggested that antibiotics were in fact effective in decreasing the mortality due to BPF.
In Promissão, all the BPF cases seem to have occurred near the sugarcane fields. If the same pattern proved true in Serrana, there could have been some underlying environmental factor that increased a child's chances of contracting the disease. But Harrison plotted each case on a map and found no geographic distinction between them. As expected, the only common factor in each household was a previous case of conjunctivitis. In going through the Serrana hospital charts, the investigators hoped they could show that a simple treatment, the application of antibiotic eye drops during the conjunctivitis phase, would prove to be a successful prevention. But they found that most of the children who died had been given this treatment, and it hadn't prevented the onset of BPF. So it obviously wasn't going to be that easy.
Fez algum tratamento? Pingou alguma coisa?
Não.
From interviewing parents, Harrison and Pontus learned that most of the cases shared a common source: a large conjunctivitis outbreak that had spread through the local daycare center a few weeks before. This gave the epidemiologists a model population to analyze. And they were able to prove that while many children had contracted BPF clone conjunctivitis, only a small minority had actually developed BPF. The vast majority had become only mildly ill or showed no symptoms at all. As before, adults were not affected, probably because they had immunity from previous exposures to common forms of Haemophilus bacteria. Despite the small percentage who became seriously ill, Harrison and Pontus came to believe that giving an aggressive course of antibiotics to all young patients with BPF clone conjunctivitis was far less traumatic and costly than the intensive care ward.
We still don't know why some children get BPF, the devastating illness, and some children can have the conjunctivitis but not go on to get severe BPF. That's not that uncommon. There are many diseases that affect only a small proportion of children who are exposed, but we would certainly like to be able to define why those children are at risk. We're also concerned about how widespread the strain is. As of now, we haven't had any additional epidemics of BPF, but we don't know how widespread the strain is throughout Sao Paulo state and whether additional epidemics might occur.
At the present time, we are alerting all of the health regions and surveillance teams in São Paulo state so that they will be on the lookout for an increase in the number of conjunctivitis cases and to detect early outbreaks, mainly in daycare centers and schools. We are also working closely with emergency rooms and hospitals so that they will be on the lookout for children arriving with fever and a history of conjunctivitis, because these could be cases of BPF.
Maria Aparecida Tobias. Tem matrícula aqui?
We will have to continue surveillance programs and even better surveillance programs, not only to detect the new diseases, the Legionnaire's disease, the Lassa fever, the toxic shock syndrome, but also to give us better information on old diseases. We also have to do surveillance because diseases are changing over time. We have mutations of organisms as the organism tries to adapt to a new environment. We have to detect those changes, whether they're a change in antibiotic resistance or a change in the expression of the disease in a person.
Sporadic incidents of BPF still occur, and cases have now been identified outside Sao Paulo state. This makes surveillance efforts critically important in Brazil and throughout the world. The story of the assault against Brazilian purpuric fever is one of determination, international cooperation, and good science. Doctors around the world have been alerted to the symptoms. And if there ever is an epidemic here or anywhere else, they will be better prepared to meet it. Like most assaults against disease, this battle remains unfinished and the victory incomplete.
For an epidemiologist, the chase is rarely over. For the most part, you simply end a particular segment of the disease history, a particular epidemic, and then you get ready for the next one. A clinician, of course, gets reinforced by every patient that improves. An epidemiologist oftentimes has to get gratification from a graph of knowing that there are people who are now protected, who will never get a disease and never know that they can have to thank that particular epidemiologist. So you get your gratification from knowing what would have happened in your absence.
Funding for NOVA is provided by the Johnson & Johnson family of companies, supplying healthcare products worldwide. And Lockheed, a bold new force in systems engineering, management, and technology services for defense, space, and industry. Major funding for NOVA is provided by the financial support of viewers like you. For a transcript of this program, send $5 to this address or call 212-227-READ.
This is PBS.